Let's now go through an example to calculate magnification using what we've learned so far. We have a sperm cell, and the length of its tail is 50 micrometers. A student has drawn the tail, and the drawing is 75 millimeters long. Our task is to find the magnification of the drawing. The first step is to remember the basic magnification formula. Magnification equals image size divided by actual size. Alternatively, we can use a magnification triangle, where each side represents a different variable, I for image size, M for magnification, and A for actual size. To calculate the magnification, we need to know both the image size, which is the size of the drawing, and the actual size of the sperm cell's tail. Next, we will substitute the values into the formula. We need to find the magnification. The image size of the tail is given as 75 millimeters, and the actual size is 50 micrometers. We will simply plug these values into the formula, and then look at the units of both the measurements. We need to ensure both measurements are in the same unit to apply the formula correctly. Since the image size is in millimeters and the actual size is in micrometers, we must convert both measurements to the same unit, which will be micrometers. To convert millimeters to micrometers, we use the fact that 1 millimeter is equal to 1000 micrometers. Therefore, to convert 75 millimeters to micrometers, we multiply 75 by 1000. The result is that the drawing size is now 75,000 micrometers. Now both measurements are in the same units. Now, we can substitute these values into the magnification formula, which becomes magnification equals 75,000 micrometers divided by 50 micrometers, and we get the magnification of 1500 times, meaning the drawing of the sperm cell's tail is 1500 times larger than the actual size of the tail. In this way, you can apply this method to solve similar problems in the future.